Good afternoon. Today we are going to begin to look at Chapter 10, which is on gene expression, transcription, and translation. Up till now, we have talked all about the cell, how the organelles are situated within the cell, how to replicate all the DNA within the cell. But one of the key questions for this class is to think about if you were to take two different cells in your body, say a liver cell and a skin cell, what is the same and what is different about those two cells? Well, the DNA is identical between those two cells. However, those two cells are going to function very differently. And why is that? Well, it's because of what proteins are currently residing in the cell and maintaining a particular function. So what determines whether or not a protein is in a cell at a particular time? Well, that is due to gene expression. Gene expression is basically turning on or off certain genes at a particular point in time. And when that happens, when we express a gene, for instance, and turn on gene expression, that means that we're going to go from DNA to RNA, and that RNA is going to get translated into protein. And that protein, then, we know is going to undergo a certain conformational structure, and that structure will determine its function and what it will then do for the cell. So this whole chapter is all about how do we transcribe genes, and then how do we ultimately translate that transcribed product into a particular protein. So in this overview of Chapter 10, we will talk through two key concepts. The first being the central dogma. I will explain what that means. And then secondly, I want to differentiate from the get-go some of the important differences between prokaryotes and eukaryotes as it pertains to transcription and translation. All right, so when we're talking about gene transcription, we are going from DNA to RNA. We call this process transcription because you are still staying in the language of nucleotides. We're going from deoxyribonucleic acids into ribonucleic acids, and we've talked all about the difference in structure between DNA and RNA, but we're still working with nucleotides. However, when we go from RNA to protein, we say that that process is translation. We are going into a new language. We are going from the language of nucleic acids into the language of polypeptides or proteins, which are comprised of amino acids. So we will go through all those steps in intricate detail, but just keep in mind, again, going from DNA to RNA we call transcription, but going from RNA to protein would be translation. And don't get that confused with when we are just replicating DNA, which was what Chapter 9 was all about, utilizing DNA polymerase. So that whole process of going from DNA to more copies of DNA is not discussed in this chapter. Again, going from DNA to RNA, we say that that is transcription because we are synthesizing a new strand of RNA, and this is going to be facilitated by an enzyme called RNA polymerase, and that will be copied from a DNA template. Going from RNA to protein, again, is called translation, so we are going to be making a polypeptide chain that's made up of amino acids, but what's our template for that is actually the RNA, the nucleotides, or the nucleic acid. And that is really our central dogma, going from DNA to RNA to protein. There's a few exceptions that have been noted in nature, but in general, this is true for every species on the planet. When we're going from RNA to protein, we need to keep in mind that there are some important steps like protein modification as well as protein folding, which I'll talk a little bit more about. So it's not just simply going from RNA to protein, but we will discuss that in more detail. So when we're talking about protein synthesis, again, what we are doing is going to be accessing a certain portion of the DNA and expressing that particular gene. So if you think back to the previous chapter where we talked about chromosomal structure, you'll remember that we talked about heterochromatin as well as euchromatin. EU, means true. So this is the true chromatin in which we're going to be transcribing the gene. So this is going to be a slightly more relaxed state than the heterochromatin where it's much more compressed. So we'll have a slight relaxing of the DNA so that things like RNA polymerase can gain access to the gene itself. So the DNA is, like I said, going to be somewhat relaxed, and we're going to transcribe that gene. We're going to make the particular messenger RNA. That's what the M stands for. We'll actually do a little bit of processing of that RNA molecule to make the messenger RNA. Once we've processed it, it's going to translocate from the nucleus out into the cytoplasm. In the cytoplasm is where we will see translation taking place. This is where the ribosomes are located. And if you remember our discussion about protein sorting, you'll remember that ribosomes are located both in the cytoplasm as well as the ER. And so depending upon where the different signal recognition particles or the absence of those signal sequences, it will either be translated in the cytoplasm or at the ER. We will then be converting from, again, mRNA into a polypeptide chain or a protein at the ribosomes, and then some modifications will take place. Particular folding will occur, and then the protein will assume its function. So one thing to note that's kind of interesting is how long this process actually takes, going from DNA all the way to protein. And we are making thousands of different proteins at any one particular time. And that process of going from DNA to protein takes anywhere from half a minute to a minute, which is utterly remarkable. Okay, so I wanted to point out a few key differences between eukaryotes and prokaryotes. Again, EU means true, so the true kernels, which means eukaryotes, when they initially looked at them, had that nucleus, what appeared to be a corn kernel. And so when you're looking at prokaryotes, they have no nucleus. And so it becomes kind of interesting to think about if we're talking about transcription and there is no nucleus, then we really never need to translocate that mRNA from the nucleus into the cytoplasm. It's already taking place where it needs to ultimately reside. And so what we find is that for prokaryotes, transcription and translation is actually coupled. As soon as there's mRNA that has been transcribed, if there is a ribosome that is docked on that mRNA, it can actually be happening simultaneously. So as soon as something's transcribed, it can begin to get translated. Now that is not true for eukaryotes, on the other hand. So as I mentioned, going from DNA to RNA, we actually make something initially called pre-messenger RNA that then gets processed into mRNA, messenger RNA. That is what then translocates into the cytosol, where translation will take place at the ribosomes. So again, for prokaryotes, transcription and translation can be coupled, but that is not true for eukaryotes. Here kind of show that a little bit more stepwise. So again, our initial stage, and this would be true for what we're looking at here, are eukaryotes because we have a nucleus. So going from the DNA, the blue and red double-stranded molecule, the first step again is to make that pre-mRNA, which then is going to get processed, and I'll describe to you all the ways that it gets processed. And this is all happening in the nucleus until what we end up with is that processed mRNA called mRNA that translocates from the nucleus out into the cytoplasm. Upon binding with ribosomes, translation can begin to occur. And you can have ribosomes in multiple locations throughout the mRNA. And so you can see here we've got our longest polypeptide chain, and so we're simultaneously making multiple polypeptide chains off of this single mRNA molecule. And the different polypeptide chains will eventually undergo some post-translational modification and undergo particular folding and then maintain its particular function. And that is shown here as well. So again, going from DNA to RNA to proteins, in which case that protein has undergone a particular folding. So one important thing to point out is that not all of our DNA gets made into protein. We've got portions of our DNA that are made up of our structural genes. We have huge portions of our DNA that actually is the regulatory sequences that determine when those genes get turned on and turned off. And we also have some genes that get expressed, meaning they get transcribed, but they never get translated. So, for instance, ribosomal RNA, which makes up our ribosomes, it's transcribed into RNA, but it never gets translated into proteins. The transfer RNAs, which are also involved in translation, they are going to be a key player in bringing the appropriate amino acid to the mRNA. Those likewise never get translated. So we do have some transcripts, some RNA that gets made but is not translated. All right, and that summarizes section 10.1 for us. I will go ahead and stop this video here, and we'll begin with section 10.2 in the next video.